Thank you, everyone. I'm based in Philadelphia. I work at Jefferson Health, Thomas Jefferson University, and I also work for OncoHost. Um, I used to work many years ago. I worked in the angiogenesis, anti-angiogenesis field, and I was then on the lecture circuit with frequently with the founder of OncoHost, a scientist from Technan University named Yuval Shaked. And about 6, 7 years ago, he met with me and they wanted, I had been involved with biomarker studies. I've worked with companies like Decipher and helped get them NCCN, CMS, FDA approval, done a lot of biomarker work with NRG Oncology. So Yuval Shaked met with me about 6, 7 years ago, asked me about an idea that they had. I didn't think it was such a great idea, and I told them I didn't think it was going to be fruitful, but I suggested that in the decision making for patients receiving immunotherapy for solid tumors, I think proteomics could be very valuable. Just to go a little macro, because it's been kind of micro over the what Chris has presented to you, we're at an incredible stage now in the field of proteomics. From an exquisitely small amount of blood, anywhere from like 2 to 200 microliters of blood, we can look at thousands of proteins. And the data you've seen is using a technology called SomaScan that looks at 7,000 proteins. We could look at 10,000 proteins. It's unbelievable. What's the reason that this juncture right now in the past couple of years has been so important is because about in the year 2000, the initial draft of the human genome came out, and that was supposed to be the operating instructions for what makes us tick and why we get disease. And it turns out it's been somewhat incomplete because although the genome codes for about 20, there are about 20,000 genes that code for proteins. There are about 100,000 different proteins. And now the ability to do high throughput deep proteomics combined with machine learning, combined with population studies. So in the UK, for example, the UK Biobank is looking at 500,000 people and they're getting blood samples throughout their lifetime on about 50,000 people. So there's been an explosion of papers where you can profile 3,000, 10,000 proteins over 50,000 people and now make predictions about all sorts of things, make predictions based on lifestyle, I mean, that proteome that they have, that you and I have, is a reflection not only of our genes, but it's a reflection of lifestyle, external factors, social determinants of health, all wrapped up in that proteome. You can show that organs, you can predict someone's chronologic age based on the proteome. You can show that different organs in the body age differently. And I think for everyone on today's call, everyone appreciates that lung cancer, even non-small cell lung cancer, let alone small cell, is a very heterogeneous disease. And we don't really understand why some people have better responses and some people don't. And why is it that two patients can have similar PD-1 expression levels and yet have a different outcome? They can have a different um, toxicity sort of profile in terms of immune-related adverse events. So I think we're at a stage now that we can use this type of information with other information that you're using currently, clinical exam, clinical factors, performance status, next generation sequencing, to add an additional element to help guide you. I'm going to ask Chris if you can go ahead. So this is when we initially started these studies, when we were developing these quote classifiers, when we looked at thousands of proteins and tried to find associations between groups of proteins in the proteome, these resistance-associated proteins, RAPs, that someone in the company called way back when. We built these models agnostically. We didn't say, oh, we're going to focus on certain biological pathways and then kind of be biased in that regard. We took an unbiased approach. And with this paper that we're happy to share with you, that got published in Journal of Immunotherapy of Cancer, what it really does is it reverse engineers what all the machine learning work that we've been doing. So we'll talk a little bit about the origin of these groups of proteins that make up this signature. We'll talk about some of the biological processes involved. We're still at the really early stages to really begin to appreciate what they reflect and what some of the potential clinical relevance is. So this may sound obvious, but it may not be so obvious that when you do a liquid biopsy, quote unquote, and you're looking at the proteome, you're taking a holistic look of a patient. You're looking at proteins that are coming from normal tissue. You're looking at proteins that are coming from tumor or different metastases. And you're looking at proteins from the immune microenvironment. And you can actually see, based on kind of the weighting of different proteins within around the 7,000 proteins. Of the 7,000, we use approximately 400 in this classifier. But you can see how many have elements in common between lung and immune, lung and normal tissue and immune, et cetera, et cetera. And this is just a kind of a gross way of looking at it. You can also look at, there are numerous ways to drill down through the data. You can look at the lung tumor proteins and see, what processes are they associated with. And again, this is just kind of our initial foray into this. I think there have been a number of papers over the past 5 years that really are trying to reduce trying at least to reduce the complexity of lung cancer to something more manageable so that we don't take a one-size-fits-all approach. But in terms of understanding what these proteins coming from tumors are doing, they certainly seem to be associated with biological processes that are associated with poor prognosis and certain known mechanisms that drive tumor progression, invasion, etc. Next slide. Okay, so when you look at all the proteins that make up our classifier, and you compare them to patients who are healthy volunteers, compare them to a group of patients who had clinical benefit, CB, and you compare them to a group of patients that didn't receive clinical benefit defined at 12 months, they kind of cluster into certain patterns. And within these clusters, there are groups of proteins. And this is just one way of looking at, what are we looking at? So we're just going to look at one of these patterns, let's say pattern E. And when you look at pattern E, where there seems to be this, not only the distribution of the particular proteins, but the level of protein expression, and the size of those bubbles relate to, meaning the weighting of this relates to, in the figure legend on the right, in terms of immune modulation, angiogenesis, etc. They seem to be higher in patients. The things that are associated with bad stuff from tumors, they seem to be associated with patients who haven't received clinical benefit from therapy. Again, all this all this analysis and all the work that Chris showed you earlier is done pre-treatment. So we're making predictions about outcome, and we're making it's the same it's the same pre-treatment sample, baseline sample in this analysis as well. Next slide. When you take a macro kind of view and you look at the proteins, there's a famous paper that's been updated 2 or 3 times by Bob Weinberg and Doug Hanahan called The Hallmarks of Cancer. What Michal Harel did in our group is she kind of put it through the lens of proteomic hallmarks of treatment resistance, and maybe it should be the liquid biopsy proteomic hallmarks of treatment resistance. And they map very nicely onto these hallmarks of cancer so that, as we look at angiogenesis, cell proliferation, extracellular matrix involvement, splicing, metabolism, all the proteins that we're finding that really define whether a patient has a probability of doing better with a checkpoint inhibitor or not doing as well, they all map onto this I would say, accepted biological processes of hallmarks of cancer, which is reassuring that what we're seeing is just not some kind of artifact. And we worked really, really hard in our program to make sure that we're not overfitting and overcorrecting. And Chris alluded to some of that, etc. So what are some of the implications of this? On the next slide, so when you start looking at some of these proteins, well, you find out that a number of them are either approved drugs. It may be for cancer. It may not necessarily be for cancer, or they may be under study under an IND. There are opportunities to repurpose potentially drugs that are being used in approved drugs that have a safety profile, et cetera, that are approved in other indications, potentially for cancer, et cetera. And this is just showing that um, there are multiple proteins within what we've studied in terms of these RAPs, where I would say conservatively around 18%, may be targetable and may be, quote, druggable. And because I think in the future, I mean, all of us right now, when we treat cancer patients, we're using some combination of therapies. Many of the newer therapies for lung cancer, whether it's a bispecific, by definition, that's a combination. Whether it's neoadjuvant, potentially with chemo or not. So by definition, as oncologists, we're in the business of using combination therapy, and I just think this sets a hypothetical stage for using proteomics as a component for drug development in oncology. Next slide. If you look at all patients and you just kind of simplify what we've done, and you look at 4 major drivers of cancer progression: angiogenesis, proliferation, resistance pathways to chemotherapy, and immune-related resistance mechanisms, and you take the patients that in this cohort that we've studied, again, this is just limiting the analysis just to reduce the complexity. You can calculate a personal score for each patient, and you could see just in terms of the normalized expression level of these proteins between on the left, the bottom where the healthy patients are, the proteins are not so overexpressed. The patients with clinical benefit, the majority of which don't have overexpression of proteins, and then the patients who didn't receive clinical benefit, you see there's overexpression number of these pathways. But just to really highlight what we've done on the right is we've taken just kind of 8 representative examples between healthy volunteers, patients who, quote, have received clinical benefit from immunotherapy, and patients who didn't receive clinical benefit from immunotherapy. And this alone shows you kind of the heterogeneity of the patients that you're seeing. So again, we're only looking at 4 different mechanisms of resistance. We could have looked at more, but just to kind of put some guardrails on this analysis. So if you look at, so you could tell that there's a difference in color, right, from expression. So if you look at the on the right side in the group of patients that are labeled no clinical benefit NCB, and if you look at patient 3, just as an example. So what you see is these pathways are not equally expressed for all these patients. Patient 3 has upregulation in an angiogenesis pathway. Patient 1 is upregulated in all of them, but is more upregulated, in resistance to mechanisms of chemotherapy and immune-related kind of modulation resistant mechanisms. And what it suggests, but by no means have we proven anything right now, but what it suggests is that you can really get down to at a patient-specific level what type of potentially therapeutic options may be beneficial for that particular patient. There may be also other things associated with this type of analysis where we can develop a frailty index, which is of interest to many people in the older adult cancer patient, et cetera. But I think the opportunity to really get to a more granular level for the heterogeneity of lung cancer and try to optimize the therapy and improve the therapeutic ratio for patients is really what excites us about this, about what we're doing in lung cancer. So we're taking just to summarize, we get a holistic view of the patient. We're able to look at tumor from obviously different sources. We're able to look at the immune microenvironment, and we're able to look at normal tissue. We think that this dovetails nicely with what's been known and developed in terms of the biology of this. We think it represents the potential future for personalized drug development for patients and ultimately to have a better future for the patients that we share in common. I don't know if there's anything after this, Chris,
Yeah,
or we're
so
going to
future
stop here.
directions, just kind of give an overview of some of the work we're doing and reading.
Yeah, so I'll just say that, with this, what we've been working on is it's kind of like with a software upgrade using the same proteins that we look at. There are all sorts of things that we're currently and actively researching. So we're going to go just kind of a little briefer through this. So we can predict, and we've done this now kind of at a population level, and we're collaborating with a number of groups around the country, in fact, if not around the world, to know pre-treatment who's going to have a severe immune-related adverse event. And we look forward to being able to predict who's at higher risk for a grade 4-5, myocarditis, colitis, pneumonitis, et cetera. So I think that in a patient with metastatic disease, you're going to treat anyway. There's no question. There are some situations where neoadjuvant therapy may be used. There are other disease sites where adjuvant therapy is being used. So the risk-benefit ratio differs in a number of situations. As oncologists, um, so we think that with your help, we're going to be reaching out to a number of you and others to try to refine where will this be useful. What's interesting, just from the biological basis of these models that we're developing, is that there's minimal overlap in terms of predicting toxicity from chemo for an individual patient, predicting immune-related, etc. So this is going to, I think over the next 6 months to a year, we'll have more information that we look forward to bringing to clinicians. I'll give you another example. Next slide. So I think everyone is familiar with ctDNA. It has a role. It's still a little perplexing where, because it doesn't explain everything. You can have patients where you see a dramatic drop, but yet they have radiographic progression. What do you do in the patient with stable disease radiographically? ctDNA is somewhat equivocal. There's no question that we have some preliminary data that doing liquid biopsy proteomics can predict 4 to 6 months ahead of time the outcomes of patients. We have a collaboration that with a group that's very active in the ctDNA as well as longitudinal monitoring of patients that we're looking forward to working with this group, and we'll find out whether this provides additional benefit to clinicians. So not only does baseline predict, but if someone obtains a 3 month or 6 month or 9 month blood test, does it help with second line treatment, third line treatment, patient with stable disease, etc. Next slide. As I alluded to, there are other disease sites that we're studying, and for example, in melanoma, in a patient with metastatic disease, one of the challenges for clinicians, many of whom on this call treat melanoma, is when to use a dual checkpoint versus single checkpoint. And sometimes more is not always better. Clearly, the toxicity is greater when you use dual IO approaches. So we have some provocative data, and we're collaborating with a number of melanoma investigators around the country. We also recently received a grant from the Melanoma Research Foundation to work with Sapna Patel, who used to be at MD Anderson, now at the University of Colorado, to analyze SWAG trials in the adjuvant setting. In renal cell cancer, one of the challenges is should you use IO versus IO-TKI. We think we can distinguish which patient's going to benefit from that, whatever combination. And we can also predict for those combinations immune-related or other adverse events to help with management and resource utilization for patients. We have work in a number of other areas. In small cell, we've had to kind of modify our foundational model, and we're working with a large pharmaceutical partner who's conducting their own studies in this space and have developed in small cell lung cancer. People have described by transcriptomics, you can define a number of subtypes, and there's a plasticity, as everyone knows. So when you take care of patients with small cell, it's a very humbling experience as they progress rapidly, and being able to pick up on this plasticity, we think we can as newer agents get developed in this space. I don't know if there's anything in this. So I think what you're seeing is where there's a plethora of tools we're looking to bring to oncologists for clinical decision support. Some of these are very well developed. Some of these are more in the emerging stages. Some need a lot more guidance from people who have a lot of patient experience to know how to best build out a toxicity predictor, etc. We don't think that proteomics solves everyone's problems and has all the answers. We think this needs to get integrated with other technologies you're using, whether it be next generation sequencing or ctDNA or anything else. And um, this is a shared journey, and we need your help, and we look forward to your advice. And I'm happy to talk with anyone offline, if they have suggestions, ideas, or would like to learn more about the journey that we're on. So thank you.